I remember writing an article for the school newspaper about how I wanted to use gene therapy to cure diseases. And I came across it when I was junior faculty, and I was like, wow, I can't believe I'm doing exactly what I wanted to do in 8th grade.
It's exciting to be in on the ground floor. I think there's no cancer that isn't on the table for potential for CAR T-cell therapy.
I remember actually being in an immunology course, and one of the lecturers was talking about how this cellular immunotherapy, oh yeah, there's some people here who are doing it. It's too big, too expensive, too boutique-y, never gonna happen. Sounds cool, but whatever, moving on. And I'm happy that that's what I ended up doing, because over a decade later, I'm here at Mass General doing that very same thing.
So, I run a lab.
I direct the lymphoma program.
And do bone marrow transplant and cellular therapy here at Mass General.
In a nutshell, what we're trying to do is engineer new chimeric antigen receptors.
Before it was even called a CAR, it was called a T-body.
We're called T-bodies.
That became chimeric antigen receptors.
Using natural ligands, using bispecifics.
Now there's trucks, armored cars, backpacks.
Combined with novel vectors.
Dual CARs, inducible CARs.
Using next-gen technologies, including gene editing.
Halins, zinc finger nuclease, CRISPR.
Bispecific T-cell engagers.
Targeted integration and targeted knockout.
Multi-specific Boolean-gated CAR T-cells.
It sounds like science fiction, doesn't it? I mean, we are literally taking the body's healthy cells and genetically engineering it to go after a cancer cell. And it involves using the B-cell apparatus, the idea of an antibody that can target a specific protein on the surface of a cancer cell, and creating that antibody out of whole cloth, saying, we want to train this antibody to attack this cancer cell. We use a virus to inject that antibody into the cell, where it can be inserted into the genome of that healthy lymphocyte. And now suddenly that T cell is equipped to destroy a cancer cell. It's science fiction meets reality, and it's curing lives. There he is.
The T cell is the most potent anti-tumor drug that there is. One T cell can kill billions of cells.
No fevers, infections.
No, like I say, I've never felt this good.
And they regulate themselves. They grow more the more tumor there is, and then as the tumor goes away, they come down and they just persist in these very low numbers, so that if you're ever exposed again, or if a tumor tries to grow again, it just, wipes it out.
That's who I'd like to meet.
The folks in the laboratory who engineer those cells, who took your cells and made them bionic.
We think that we've learned a lot from our initial experience, but there's more to learn. We're trying to understand better how the immune system works. What happens to the T cell when we put CARs into it and we change its whole signaling pathway? We've diverted so much of what a T cell naturally does to make it do what we want it to do. And, we're trying to test the limits. How much can we really manipulate the T cell and still maintain control of it?
So based on the in vitro data that I have shown you, we then proceeded to test our anti-multiple myeloma CARs also in a mouse model. The BCMA CAR-treated group and the Tripro CAR-T-treated group responded with very similar kinetics.
Tripro looks awesome.
It works, definitely.
My goal is to be able to treat at least 3 cancers in clinical trials in the next 3 years. So we're starting with lymphoma, both B-cell and T-cell lymphoma, and multiple myeloma, and glioblastoma, which is the brain tumor. If you think about the timelines, like how long does it take to test a new drug? It used to be measured in decades. In CAR T therapies, that whole process can take 18 months. We have now Tripro. I didn't think that this would be so fast, that it would work so well. We have a dual specific that's a natural humanized protein. The other thing is, all the other ones are mouse-based and patients get immune responses to it. Ours is all human. The challenge will be getting the funding for it, but we could have 3 trials open in the next 2 years. So get ready for the ride.
CAR T-cell is a way of engineering someone's own immune system to identify the cancer that may have not responded to anything else, and to completely eradicate it, to cure it.
It's really exciting. I try to be a little bit conservative in my thinking and, be reality based, but the truth is it's amazing. But when I started working as a fellow in oncology, we were never allowed to use the word cure because it felt like it would give false hope. And now we use the word cure.
It's cure with a capital C. We've seen patients with lymphoma that was resistant to every type of chemotherapy available. These are patients for whom their life expectancy is measured in a few months. And with a single infusion of their own engineered cells, we look 28 days later and see complete remission of their disease. So here's what we started with. The left side of the chest completely filled with cancer. And so that's right before the CAR T cells. 28 days later, after that single CAR T cell infusion, we see a complete remission. No evidence of persistent lymphoma where we had it in the chest or the abdomen. Just a normal brain, normal kidneys, normal bladder. What a difference 28 days can make. One of the great things about Mass General is at the very highest levels of leadership, they recognize that we have a shared vision of innovation, driving the optimal advances in clinical care, all to help the patient in front of us.
There's a culture behind that too. And no one rogue person is going to come in and modify that culture, because I think it's something entrenched in a place like MGH and protected and fought for.
I trained as a physician and as a scientist, and my whole reason for being was to be able to apply science to treat patients.
At Mass General, they've said we are all in. What do we need to do to develop this technology and bring it to the care of our patients?
Like I said, I've never felt better in my life than I do right now. Yeah. And I just can't thank you enough.